Hi, I'm Jay Phelan. Let's talk about some biology. We're going to talk today about linkage and recombination, and this fits into the category of extensions to Mendel, places where Mendel set us on our pathway to understanding inheritance, but because of things that weren't exactly as he would have predicted, it's driven us to figure out kind of a more nuanced, accurate view of how inheritance works. Now, Mendel, remember, his conception of inheritance was based on his studies looking at 7 characters in pea plants without any knowledge that chromosomes existed. So he assumed that genes were just these free-floating hereditary factors within a cell. There were 2 copies of each, 2 alleles, and 1 copy got passed down to each gamete. That was his law of segregation. This is very important. And as a consequence of this, that each trait had 2 alleles and they were floating around, he expected independent assortment. What you inherited for one trait should not influence what you inherit for another trait if we were to look at 2 of them simultaneously. Therefore, if you were to cross individuals that were heterozygous for each of 2 traits, you could still treat each trait as if it was an independent thing. And that leads us to an expectation of 9:3 to 3:1 of phenotypes that we would get from that double heterozygous cross. Now, in his simple view, if we were to look at color, say yellow or green with yellow being dominant and round or wrinkled with round being dominant, if we crossed true breeding individuals, yellow and round and green and wrinkled, we'd get all yellow and round F1s, but they'd be heterozygous for both traits. Then when you do the cross of those yellow, round, doubly heterozygous individuals, you do a big 4x4 Punnett square. But what you do when you set up the gametes for the female, the gametes for the male, if you're doubly heterozygous, big Y, big R, little r, you'd have all possible gamete types. You'd have a big Y and a big R, or you'd have a big Y and a little r, or you'd have a little y and a big R, little y and little r. So all of those go across the top. Same thing for the male, and then you figure out where do they fit in. When you come up with all the 16 different combinations and you look at, all right, if it has either homozygous dominant or heterozygous, it's still going to be dominant for the trait. We see that we expect 9 out of the 16 would show the dominant phenotype for both traits. 3 out of 16 would show the dominant for one, recessive for the other. 3 out of 16 would show the recessive and the dominant. And 1 out of 16 would show the recessive for both. We expect that in those 4 gametes that are produced, they're produced in exactly equal numbers. And when we have 4 possible gamete types here from the female, 4 from the male, we expect the probability of any combination to be equally likely. And that's an important thing to view. Now, what if Mendel had been working with a different species? Around turn of the century, a little bit later, after Mendel had died and after chromosomes had been discovered, another researcher, a guy, Reginald Punnett, famous for his Punnett square, he started looking at heredity, but he was looking in a different species of pea plants, sweet peas. And he looked at color, he looked at purple, and he looked at the length of a component of the plant. So when Punnett did the cross, his double heterozygotes were purple and long, but they were big P, big L, little l. When he crossed those, we'd expect the same thing that Mendel had. We'd expect the purple and long to be 9, purple and round to be 3, red and long to be 3, and red and round to be 1. He looked at 381 plants in one of his experiments that he reported on, and what he found when he did this cross, so I've just set it out a different way here. He had red and round, purple and long, and when he crossed those, the dominant won out for both of them, so they were all purple and long, but he crossed the F1s, so they are the doubly heterozygous individuals, and you would expect 9:3 to 3:1 of the genotypes, big P, something else, big L, something else, big P, something else, little l, and so on. If you put numbers on the 381, our expectation would have been 215 out of 381. That's just 9/16 of 381. But what he observed was this. At first glance, it's like, all right, pretty close. But you look closer and you see something's not right there. 284 instead of 215, 21 of the individuals that had one of the dominant traits and one of the recessive traits, something's not right there. And then red and round as well, more of those. It seemed that he got more double dominance. The purple and long, more of the double recessives, red and round, than he had expected, and conversely got fewer of the other individuals. This is an important take-home message that often Mendel's second law, independent assortment, just doesn't hold true. It's not what we observe. If we cross individuals that are both heterozygous for two different things, we don't see a 9:3 to 3:1 ratio of the phenotype. So Punnett saw that, and he was trying to figure out, why is that? What's going on here? So he looked at it, he thought, all right, the question to answer is, why are there more double dominants? Why are there more double recessives than we expect? There are a couple different options for what that could be. The first could be maybe the F1 plants weren't making equal numbers of all four possible gamete types. Or maybe they were making equal numbers, but they weren't coming together with the same probability. So Punnett was kind of interested in that idea. Around the same time, a guy named T.H. Morgan was working at Columbia. He's a very famous guy. He worked with fruit flies, but he was interested in the same questions. Doesn't matter if you're looking at plants or animals, you can still study inheritance. And Morgan, studying Drosophila melanogaster, he did these experiments for which he ultimately got the Nobel Prize in 1933. He said, I'm going to look at the trait of eye color, so it's either purple or red, and I've got PR for purple. That was initially identified as a mutant. PR plus, the plus is used historically to designate the wild type, the most common one that we see. And then for wings, VG stands for vestigial, and VG plus is a normal wing type. So he crossed purple and vestigial, homozygous recessive for each of these two traits, with red and normal, homozygous dominant for each of those. The F1s is going to get the same thing that Mendel got, same thing that Punnett got. Here's a picture of the vestigial wings. You can see that when they develop, they're just kind of shriveled up. They can't fly, and it's easy to identify just looking at them with the naked eye. And here is the purple eye and the red eye, just so you can get a sense of what they look like. So in its cross, as expected, we get red and normal. The wild type wins out in each, but they're going to be heterozygous for each. So, okay, nothing different here than what we've already looked at. But here's what Morgan thought. He thought, forget about crossing the F1s. What if we test cross the double heterozygous F1s? Now remember what a test cross is when you take some individual that shows the dominant trait, you don't know what the full genotype is, and you cross it with an individual that is homozygous recessive. And that way then from the offspring we could figure out the genotype. But he said, let's test cross these F1s that are heterozygous for each of the two traits. And I'll show you what happened. Think about the gametes that they're producing. The F1 double heterozygotes, PR, PR+, VG, VG+. The test crosser is going to be the recessive for both, PR, PR, VG, VG. So we know the test crosser gametes. Mendel's law of segregation, they're going to put in one PR and one VG in each, but all they have are those. So it's every gamete is always going to be the same. For the heterozygous F1, it's going to be a PR and a VG, or it's going to be a PR and a VG+. It's going to be a PR plus and a VG, or PR plus and a VG plus. Those are the four options that they could put together. So we've got four options for the one individual. We have one type of gamete for the test crosser, and then we look at what are the genotypes. So I've drawn them in here with the test crosser gamete that comes together at fertilization in green. It's always the same thing. And then for the other individual, it's going to be either the PR or the PR plus, the VG or the VG plus. I drew those in blue, and we expect equal numbers of these four types of offspring. They're all always getting the same thing from the test crosser, but from that F1 individual, they're getting one of these four types of gametes that it can make. And since we started looking at Punnett squares, we always said, all right, they make all the different gametes in equal numbers. So we would expect 1 to 1 to 1 to 1. They should all look the same. So this is a take-home message. This is important. If you test cross the F1 offspring of a dihybrid cross, dihybrid meaning they're doubly heterozygous, it's going to show us the proportion of each gamete they're producing. Now let me go back for a second. When we look at what these offspring look like, if they show purple, it means that they got a PR from the F1 because they always get a PR from the test crosser. If they show red, it means they got a PR plus from the F1 because they always get a PR from the other one. So that's interesting because that means that we can look at the fly. If it's got purple eyes, we know that the gamete was a PR. If it has red eyes, we know that the gamete was a PR plus. Same thing for the wings. If it has vestigial wings, we know that it got a VG from that F1 individual. If it has normal wings, we know it got a VG plus. And this is really clever insight. It means that by looking at the fly, what color are the eyes, what do the wings look like, we can tell what gamete the F1 contributed. And what's cool about that is that gametes and genes, they're tiny. We can't see them. But now by looking at a physical trait, we can tell exactly what gametes were being produced. Our expectation was 1 to 1 to 1 to 1. But now we just look at the offspring and we can find out, are they producing gametes in that proportion? And so you have to think about this and contemplate. This is a really clever experiment. I really think this is as creative as any beautiful piece of music. The idea that you take a doubly heterozygous F1 and you test cross it. So now physically you can see something that is too small to be able to see. So there's T.H. Morgan, smart guy. Anyway, all right, so we look at Morgan's results. So he's expecting purple vestigial, purple normal, red vestigial, red normal, those 4 types. We said in equal numbers. Well, what did he find? He found, when he looked at like 2,800 fruit flies, it's nice, you can get lots of offspring when you do crosses with these. He got tons of the purple vestigial. He got tons of the red normal. You got very few of the other 2 classes of offspring. Certainly not 1 to 1 to 1 to 1. You look at those, you know something's going on. What does it tell us about the gametes that are being produced? Well, look at the blue ones here. 1,339 means, out of the 2,839, means that PR, the blue PR VG, a lot of those gametes. The PR plus and the VG plus, a lot of those gametes. Not very many of the PR VG plus. Not very many of the PR plus VG. So the gametes were not being produced in equal numbers. Way more PRVG and PR plus VG. Also, another thing, a little bit more subtle, is that you get about the same numbers of the PRVG, PR plus, VG plus, and you get about the same numbers of the other 2 together. So Morgan had to figure out, all right, what's that telling us? What's going on? So you might do another cross. Think about this. Our original cross, they were red and normal, purple and vestigial. And you cross them, double heterozygous, they show the dominant for both. What if instead of the 2 dominant traits, red and normal, and purple and vestigial, what if we move the purple over here with the normal wings, and we take the red over here with the best up wings? What if we said this? We say purple normal, red vestigial. From a genotype perspective, all right, it's just little p, and VG plus, and so on. So when you cross them, you're still going to get all red normal things. The dominant and recessive interactions between them are the same. The genotypes are still going to be double heterozygotes. Okay, but then take those double heterozygotes from the F1 generation, and what are we going to do? We're going to do that great cross, the test cross. So we test cross them, the double heterozygote. It looks the same as his first experiment. But here we created those double heterozygotes by parents that had sort of the 2 traits in different combinations with each other. So when you look at this one now, same thing. I've got the green test crosser gametes, and I've got the blue gametes from the F1 individual. We would expect them to be in all equal proportions. We would say purple vestigial, 1 purple normal, 1 red vestigial, 1 red normal, 1. Instead, the observed showed the same pattern, but reversed now. We saw very few of the double recessive, very few of the double dominant. We saw lots of 1 recessive, 1 dominant, or 1 dominant, 1 recessive, by starting with the different material. So Morgan came to the conclusion, I think something's going on. I think that the genes for these 2 traits, I think they're on the same chromosome. And that's causing them, depending on which ones happen to be close together genetically, it's causing them not to assort independently. It's causing them to want to stick together. We can look at his first cross in a different way. We say, all right, red normal time crossed with purple vestigial. But now I've put the PR plus, PR plus, VG plus, VG plus. I've put them with a little line to stand for the chromosome. So the PR plus and VG plus are stuck together. They're on a chromosome, a linear strand of DNA. For the test crosser, PR, VG are stuck together, PR, VG are stuck together. When we look at what the gametes are, they're still going to be PR plus, VG plus for the F1. With the test crosser, is going to be PR, VG. But the F1 is going to be like this. It's going to be a PR plus PR, VG plus VG, but the PR plus and VG are going to always want to go together. So I've drawn a little dotted line around and a tail on it. If it makes a sperm, the PR plus and VG go together. The PR and VG go together. Typically, if we have a double heterozygote, we'd say, oh, all 4 combinations are possible. PR plus, VG plus, PR plus, VG, PR, VG plus. So we look at that sort of visualization of the cross, that a PR and a VG are stuck together. If this is the case, then instead of 4 types of gametes, we expect just 2 types, the PR plus and VG plus, the PR and the VG. And they're always going to go together. So equal numbers of each. But when we get, during the cell division, during the production of the gametes, we're going to have those 2 pairs always together. So in his first test cross, we could say, all right, so we've got the red normal, and you should try to draw this out. What does it look like? So these are going to be, remember, PR plus, PR, VG plus, VG. So they're showing red and normal, but they're double heterozygotes. The test cross are going to be PRVG, PRVG. But you want to draw them together. So when you put the expected results now, you're going to get either the PR plus and VG plus together or the PR and the VG together. So this is what the F1s would look like from Morgan's first cross. They're double heterozygotes. It's still PR, PR plus, VG, VG plus. But we expect only PR-VG gametes, only PR plus VG gametes, only 2 types of gametes. Therefore, when we do the test crosser, half of them should show recessive traits for both, half of them should show dominant traits for both, and that should be it. Unless there's some crossing over. Now we know from the way meiosis works that sometimes when we get the homologous pairs of chromosomes coming together, I've got drawn in blue, that PR and VG, they're stuck together on the chromosome, the PR plus and the VG plus. But if there's a break in the chromosomes, and then they reattach such that the PR on top is now curves down and is attached to the VG plus, there's going to be a simultaneous, also linear strand that's the other one, PR plus and VG plus. Every time there's a recombination event between the 2 genes, we're going to swap them, and then you're going to end up with PR and VG plus, or PR plus and VG together. Doesn't happen very often, and the closer together they are, the less likely it is to happen right between there. But every time it does, then you're going to get some gametes that are not the ones we expected, the PR and VG, the PR plus and VG plus. So to some extent, we expect not just 2 different types of offspring, we expect 4 types, but we expect a lot of 2 types and a few of the others. So here is a visual representation of what the gametes would look like if a crossover event happened. I just finally have shown that the wild type PR plus could be paired up with the vestigial wing allele. The PR, the purple eye color, could be paired up with the wild type for the wings, and we'd expect equal numbers of those 2. So our final take-home message here is this. When you have linkage, doubly heterozygous individuals are expected to only produce 2 types of gametes instead of the 4 that we would normally get, unless there's a recombination event. And every time there's a recombination event, we're going to create a small number of the other types of gametes that have the things that are on the opposite chromosomes that they inherited from the mother and from the father. In another video, we're going to look at some examples. This can be clarified when you look at the numbers. How often does recombination occur? How is it related to how far apart they are? But we'll stop here just to have demonstrated that we don't always see independent assortment. And now if we view it from the perspective of alleles exist on chromosomes, and if the alleles for two different traits, the genes for those traits are on the same chromosome, then we don't expect them to assort independently. That's it.